Respected madam, my name is Teja Malluvalasa. I'm studying in third year B Pharmacy, and my presentation is about the applications of genetic engineering. And coming to genetic engineering, it is also called as genetic modification or gene manipulation in the direct manipulation of an organism genes using biotechnology. It is a set of technological and to change the genetic makeup of cells, including the transfer of genes within and across species boundaries to produce improved or novel organisms. And mostly genetic engineering is based on the recombinant DNA technology. It is that is used to cut the known DNA from one organism and introduce it into the another organism, thereby genotype manipulation takes place in the recipient and then known to form a new recombinant product. And coming to applications in pharmaceutical industry and medicines, with the help of genetic engineering, several of medicine products, insulin and human growth hormone were first commercially accessible, produced and achieved from recombinant DNA, recombinant technology, that is E. coli bacteria. For example, like therapeutic product like insulin, and it is applicable in diabetic treatment, and growth hormone, it is applicable in the treatment of hormone deficiency. And coming to the applications in food industry, with the introduction of genetic material of plants or animals in the market, genetic engineering finds widespread application in food industry. Genetically engineering foods decrease the need for insecticides and pesticides as the yield post resistant and drought resistant crops and upsurge production. It also has potential to create new or more productive strains of from animals for meat and milk production. Next, coming to applications of environment. Genetic engineering is developing the enormous perspective of microorganisms, plants, animals for the restoration of the environment. Genetic engineering is aggressively tangled into the development of various microorganisms and bacterial catalysts for remediation of polluted atmosphere and expansion of eco-friendly produces life like recombinant strain for biophysical fabrication, etc. There can be genetically engineered to generate substantial extended period of environment and economical benefits. According to researchers, viral orders can be introduced into conveyed in plants as they could restrict with the life cycle of the similar or a strictly located challenging virus. Through this, they alter resistance the infection. For instances, for first trees, fruit trees, code protein gene of plant PPR utilized in transgenic proteins plants has delivered high levels of resistance besides the virus. And now coming to application in crop improvement through transgenesis. The attention of genetic engineering is majorly towards crop plants. Efforts are being taken to increase the tracts of plants. Transgenic plants are established for causes like expansion in the agricultural marketable value plant by investing gene and understanding the purposes of gene. Several genes can be joined with crops to produce required positions like faster growth, improved for values in maize, edible plant vaccines, fast ripening process, and fast ripening. And now coming to application in trait improvement of plants through transgenesis. Genetic engineering includes the outline of transgenic animals to advance the trait of transgenic animals, elimination of healthy cells from a female host and fertilization in the laboratory. Transgenic animals therefore largely direct trait of the presented gene. Transgenic animals are also generated to study the purpose of different gene to improve treatment of a disease. And there are various genetical applications like genetic engineering of bacteria and fungi, using of drugs, vaccines, antibiotics, food products, etc. In example, cheese, metabolism and biofuel production, human gene delivery systems. And coming to genetic engineering of animals and plants, biofarming and products like drugs, vaccines, proteins, using animals to make drugs, transgenic crops, as we spoke like transgenic crops like corn, soybean, cotton. And coming to genetic engineering of humans, coming to germ cells versus somatic cells gene therapy, and in vivo gene therapy, ex vivo gene therapy, and like this, based on various applications. And nowadays genetic engineering has a various widespread of applications in biotechnology and in the normal human processes. And coming to preparation of vaccines using recombinant DNA technology, a harmless form of a pathogen made from recombinant DNA tells your body what a specific pathogen looks like. If your body knows what it looks like, it can fight and can be used to produce the antigens needed in a safe and controllable way. The use of genetically modified yeast cells to produce a vaccine against a hepatitis B virus has been a major success story. For example, how the RNA vaccine works. Scientists take part of the virus genetic code and turn into a vaccine that is injected into the patient, and the patient enters the cell and tells them to produce the coronavirus spike protein. The body immune system reacts, produces antibodies and T cells to destroy cells with the spikes proteins. If the patient later catches coronavirus, the antibodies and T cells are triggered to fight the virus. This way, the vaccination helps the person to fight the virus, and various other modifications took place in case of insulin, and various other applications are approved in genetic engineering. These were the references which I took part and extracted the knowledge for the seminar. Thank you, ma'am.